Hello students, this is Molecular Basis of Inheritance part 10. In this session we are going to talk about the packaging of genetic material. So the length of the DNA molecule or double helical structure of the DNA in a typical mammalian cell is approximately 2.2 meters, and normally a cell is having a size approximately 10 to minus 6 meters. So how this long DNA molecule can be then get accommodated in such a small nucleus? It therefore must be condensed, coiled, and supercoiled to fit inside the small nucleus. So the condensation of the DNA molecule or condensation of the genetic material, it occurs in the prokaryotic cell as well as in the eukaryotic cell. So in this slide we are going to discuss about the packaging in the prokaryotic cell. So you have studied the cells of prokaryotic. You have studied the different cells, right? In the 11th standard you have studied the detailed structure of prokaryotic cell and the eukaryotic cell. So I'm not going to discuss the structure of the cells here right now. So now this is the diagram. In this diagram you can observe this is a typical prokaryotic cell, and it this part we are talking about now, this is the chromosome, or this part is called as nucleoid region. Okay, now this part we are going to discuss in the packaging in the prokaryotes. So in prokaryotes, like in the E. coli cell, that is in the Escherichia coli, the cell size is almost 2 to 3 micron long, and they don't have well organized nucleus, as you have studied the eukaryotic cell structure. So in the eukaryotic cell they have a nucleus where the chromosome or the genetic material is fitted inside, right? So they don't have well organized nucleus, so you can see there is no a separate compartment for the DNA. So these cells are without nuclear membrane and the nucleolus. So as you can see, there is no presence of nuclear membrane and there is no presence of nucleolus. So the nucleoid is small, it is circular, it is highly folded, naked, and it's a ring of DNA. Okay, so nucleoid, now this structure you are talking about. So this nucleoid is, it is small, it is circular, it is highly folded to fit inside the bacterial cell. It is naked ring of the DNA. So that's all about the structure of the prokaryotic cell and its DNA. So here we are going to talk about the packaging of genetic material in the prokaryotic cell. So this is the structure of the nucleoid of E. coli, Escherichia coli, and you can see this DNA or the genetic material of E. coli. Now these are supercoiled uh, DNA structures. Okay, so these are the thread-like structures which are coiled and supercoiled, and then they are fitted inside the cell, that is in the prokaryotic cell. In this slide, we are going to talk about how the DNA packaging it occurs stepwise, which is shown in the diagram. This is circular unfolded chromosome. Uh, you can see here the size 350 micron. Now, when it gets folded, now this is the folded chromosome. It contains 40 to 50 loops. Now, this structure, now this is a circle, right? This is completely circular and unfolded DNA. Then it's get folded into 40 to 50 loops. Okay, and this is again in the third step or in the third stage, it again gets supercoiled. You can observe now each loop is again supercoiled. Okay, so each loop is independently supercoiled in the next form. Okay, and another thing is when the chromosome it gets partially unfolded. Now this is RNA cleavage, partial DNase digestion when it occurs. Now these are the groups of enzyme, like the partial DNase or the partial RNase digestion when it occurs. So you can observe the structural change. Okay, so there is structural change. It gets partially unfolded here, and sometimes it is getting partially uncoiled. Okay, now this is partially uncoiled chromosomal structure. So these are the DNA packaging steps which are present. Now why it is required, why I have already explained it, that the DNA length is about 2.2 meters and the size of the nucleus is 10 to minus 6. So you can just imagine the length of the DNA, it is very large. So this long DNA molecule has to be accommodated inside the prokaryotic cell, which is very small in size. So it must be condensed. Now there is condensation, there is coiling, and this is called as supercoiling. Okay, this is condensation, this is coiling, and this is supercoiling, which occurs for the packaging of the DNA in the prokaryotic cell. So in this slide, you can observe that how the partial RNase and partial DNase, they give structural changes to the genetic material. Okay, so this is the circular DNA. Now it is getting folded. So this is the RNA protein core. Okay, in the next step, it gets supercoiled. Now this is the supercoiled DNA structure. Now the partial RNase reaction, the structural deformation you can observe, and there is partial DNase reaction. Because of this, the nick formation it occurs. Now you can observe there is nick. Nick means a cut. Okay, so that cut, when it occurs at some site of the DNA, the strain or the stress get relieved. Okay, so that stress get relieved, and because of that, the structural deformation or structural change get formed in the structure of the. So with the help of animation, we will try to understand that how the circular chromosomal DNA it gets changed. Okay, how it gets supercoiled. So the negatively charged DNA, now this is the negatively charged DNA, which is circular. It reduces its diameter, it gets reduced or get folded. Now these are the foldings, or these are the loop domains. These are the DNA binding proteins which are helping the DNA to form the loop. Now this each loop, just observe this animation. Now this each loop, it gets folded again. So there is supercoiling of the DNA. Okay, so near about 40 to 50 loops they are getting formed. Now loops are formed due to the RNA connectors. So there is loop formation because of the RNA connectors. Now this is the nucleoid which is present inside this bacterial cell. So each domain is coiled and supercoiled, thereby reducing the size down to the 2 micron in diameter. So I hope you have understood how the genetic material it gets packaged inside the prokaryotic cell. The enzymes like gyrases and topoisomerases, these are the enzymes which are very much helpful for the supercoiling of the genetic material. So that's all about the prokaryotic packaging of genetic material. We are going to discuss the eukaryotic genetic material packaging in the next session. So that's all for today's session. Thank you.